Hello, welcome to our video on fertilization and organogenesis. There's a previous video that we uploaded on spermatogenesis and oogenesis, that is generally gametogenesis. If you have not watched the gametogenesis video, you do well to watch it before this one because somehow it would have an impact on your understanding of fertilization and organogenesis. Now, straight to business. What is fertilization? Fertilization simply involves the fusion of the male gamete, which is the sperm, and the female gamete, which is the egg or ovum, to form what is called a zygote. Now, ordinarily, the sperm and the egg are known to be haploid, whereas the zygote is diploid. So it means that fertilization affords the gametes an opportunity to recombine the chromosomes. Because ordinarily at the point where, according to the other video now that you must watch, according to that video, during gametogenesis, the gametes, that is the sperm and the egg, actually come from diploid cells. So it means that in the formation of these gametes, diploid chromosome numbers, that is the 2n, becomes reduced to the haploid number n. So chromosomes that otherwise would have been paired in the original cell became single upon formation of gametes. So when two gametes come together now, those chromosomes that were originally paired will end up pairing up again. So we say that fertilization brings or affords an opportunity for homologous chromosomes to recombine. Then apart from that, another important reason fertilization should take place is without fertilization, the development of an ovum usually does not continue. According to that same video, we said it there that when an ovum develops, it does not become an ovum until fertilization takes place. Instead, it stops at what we call the secondary oocyte stage. So for a secondary oocyte to progress to become an ovum and beyond, there must be fertilization. So those are two important reasons or two important benefits of fertilization taking place. Now, this fertilization, from what we've said, involves the sperm coming to fuse with the egg to form what is called a zygote. So it means that N combines with N to give 2N. Now, for a sperm to fuse with an egg, the egg originally, upon the process of ovulation, is released into a part of the woman's reproductive system that we call the fallopian tube. So the fallopian tube or oviduct is where ovulation occurs into. So that as you have the fallopian tube like that, you have the egg somewhere there. The secondary oocyte, it has been released by the process of ovulation. Now, for this egg to be fertilized by a sperm, first, the sperms will need to be introduced by a process that we call coitus, which is the sexual act. So by coitus, sperm cells are introduced into the female reproductive tract. And from there, the sperm cells have the duty of swimming all the way until they reach the eggs. Now, it is true that in some lower organisms, there's enough evidence to show that the sperms actually get attracted to the eggs. But of course, not in humans. In humans, the evidence that abounds show that the sperms would need to swim. Now, interestingly, if you look at the female reproductive tract, you realize that there are two fallopian tubes. Now, just imagine that ovulation occurred on this tube. It means that all of the sperms that have gone in this direction have missed, as it were. Then for the ones that go in the right direction, many of them end up not even reaching the eggs. Then, of course, there are those few ones that get to reach the egg, but there's just one lucky one that would fertilize the egg. So usually, one egg can only get fertilized by one sperm. Now, the sperms flow, or like we said, swim up to the fallopian tube where they meet the egg waiting. Now, these are different sperms. I've tried to illustrate in a diagram here. These are different sperms. strolling or swimming towards an egg. So that's the egg. We're not showing all of it. All right, so we're just showing part of it to highlight the important point here. Now, as these different sperms run towards the egg, even before they get to the egg, they would need to undergo a process called capacitation. Capacitation is like building the capacity to fertilize an egg. Without fertilization, a sperm cell, even though it meets an egg, or even if it eventually meets an egg, will not be able to fertilize it. So it must be capacitated first, and this capacitation involves modification of its glycoprotein coat that covers the head. If you watch that previous video, you see the structure of a sperm cell. So the head has glycoprotein coat on it that becomes modified, and then also the membrane of that head becomes more permeable to calcium ions. Now, these calcium ions that are going to flow, this influx of calcium ions into the head is very important for the next thing that happens after capacitation. But of course, the first thing to happen is capacitation. And when should it happen? Even before the sperm cell reaches the egg. Now, upon reaching the egg, a second process takes place, and it is called the acrosome reaction. The acrosome reaction. Now, this acrosome reaction, like I said, is triggered by the influx of calcium ions. Now, when the calcium ions flow into the sperm's head, and that head reaches the vitelline membrane of the egg, the membrane, the plasma membrane of the sperm's head fuses with the vitelline membrane of the egg, and then that fusion will be followed by a penetration. So the sperm has to penetrate, has to enter inside the cell for fertilization to take place. But for it to even enter, its membrane must fuse with the vitelline membrane, and then it is able to penetrate. But after the sperm penetrates, now here's the sperm, it has entered the egg. After it has entered the egg, certain things must take place. For example, the tail of that sperm will be shed. Of course, it no longer needs the tail, so it can shed the tail. That's one interesting thing that happens after the sperm enters the egg. So the tail is not shed outside but inside. And then also, after the sperm enters the egg, this vitelline membrane that was receptive to the sperm, we said that the membrane, the plasma membrane of the sperm's head fuses with the vitelline membrane of the egg, and then the sperm goes in, it rolls in, as it were. Once that sperm goes in, the vitelline membrane will undergo modification. That modification makes it unreceptive, more like it becomes a barrier, it becomes a brick wall, so that no other sperm can enter. That's what makes it possible for just one sperm to fertilize an egg. Else they would all rush in, and there becomes plenty of chaos. So only one sperm can enter an egg. Once that sperm enters, we said the vitelline membrane will become modified. The modification that occurs in the vitelline membrane is summed up in the term cortical reaction. So the cortical reaction is a term we use to describe the many changes that occur in the vitelline membrane, preventing other sperms from coming in. Again, that still has to do with calcium ions, and then what we refer to as the cortical granules. So there are cortical granules within the egg, and they play a very vital role in what we call the cortical reaction. So it means that in fertilization, or for fertilization to take place, three important things that should occur are capacitation, the acrosome reaction, and then the cortical reaction. This prepares the sperm for entry. This occurs at the very point of entry to facilitate entry, and then cortical reaction prevents further entry, prevents the entry of further sperm cells. So now that the sperm is in, the tail is off, this is just the head. And what that head contains, because the acrosome is gone, that acrosome, if you watch the previous video, we said it contains enzymes, hyaluronidase, acrosome. All of those enzymes pave the way. They clear the way for the sperm cell to enter, so that what is left of this sperm cell is nothing but the nucleus in the head. So that nucleus of the sperm cell will fuse with the nucleus of the egg cell. And when those two nuclei fuse together, we say that the egg cell has been fertilized. And the number of chromosomes present in the egg cell would no longer be 23, but 46. So which means a zygote is actually not two cells. Instead, a zygote is one cell. A zygote is an egg cell that has been fertilized. So at the end of fertilization, yes, we get a zygote that is diploid in terms of number of chromosomes. And of course, that's the very end of fertilization. Here I've shown an algorithm. I've shown that from egg to organism, there are 5 important changes that take place. The first one we just talked about, that's fertilization. It achieves the conversion of an egg into a zygote. What happens to the zygote is what we'll talk about after now. So how the zygote becomes what we call a morula, so the process of cleavage, we'll talk about it. And then how the morula becomes the blastula, that process is called blastulation. And then the blastula becomes the gastrula, that's gastrulation. And then by organogenesis, the gastrula begins to develop the different organs we have, including the eyes with which you're watching this video. All right, and then over time we get the full organism. So I'm going to talk about these processes in very detail, though. Yeah, I mean, not in the same or to the same extent of details. These ones, the first two here, that is 2 and 3, I mean now, because we're done with 1. 2 and 3 will get more attention than the others. Gastrulation especially, not so very important in terms of the outline, because as we make this video, I have an outline. I have a course outline in mind. That is the AEB 122 outline for Unibed. So the AEB 122 outline is like my guide. So I'm trying not to go out of it so that I don't give you more details than you need. So that is the much we'll take for fertilization. Now moving on to the second process, which is called cleavage. Cleavage, from what we're seeing here, means the conversion of a zygote into a morula. Now, cleavage is a type of cell division. Just as you have meiosis, you have mitosis, there's also what is called cleavage. So what is cleavage? Now, cleavage, like I already said, is a type of cell division, but it differs from meiosis and mitosis in that, in the case of meiosis and mitosis, if you have, this is mitosis for example, look at mitosis, you have one cell like this. This cell will eventually divide into 2 daughter cells that are of the same size as the parent after they grow, excuse me. So this is the parent and these are the daughter cells, and you see that their sizes are about the same. So which means that when mitosis takes place, nuclear mass and cytoplasmic mass will increase. That is when this one, of course, ordinarily if you were to use your eyes to divide this, you will not get 2 cells that are this big. But how do the offsprings or the daughter cells become this big? Because there's an increase in their nuclear mass and cytoplasmic mass. But in cleavage, you have one cell that is this big. It will divide to form 2 cells. Now see the 2 cells. Number 1 difference, the 2 cells are not separate. Here they are separate, you have them as daughter cells. But here the 2 cells are still together. That's the normal thing, the usual thing that happens in cleavage. Now apart from the 2 cells being together, their total size, their total cytoplasmic content is the same as that of the parent. Meaning, if the parent cell, assuming, contains 2 liters of cytoplasm, then these 2 cells put together will contain a total of 2 liters of cytoplasm. In other words, the key difference between cleavage and mitosis is, in mitosis there's an increase in cytoplasmic mass after division, but here no increase in cytoplasmic mass. What is more, in mitosis, if these two daughter cells were to have their own daughters, the daughters will still have the size of the parents. But here, if these two were to divide further, you may now have something like this. So these three are the same size. This goes that way, that goes that way. So that eventually, how many cells do I have here? 4. But their total size is still the same as the original zygote. Unlike here, you have a larger number of cytoplasm, a larger volume or mass of cytoplasm. But here, the overall cytoplasm is the same. So it means that these 4 daughter cells have approximately 25% of the cytoplasm of the original zygote. So this is the difference between cleavage and mitosis. Now, having mentioned the difference between cleavage and mitosis, I'll tell us about types of cleavage. But before telling you about types of cleavage, I should tell you about types of eggs. Because if you know about the types of eggs that we have, it is easier to appreciate the types of cleavage. So see types of eggs. Eggs may be alecital or microlecital or mesolecital. Mesolecital is also called oligolecital. And then there is macrolecital. For a truth, some persons use oligolecital as an alternative term for microlecital. So leaving oligolecital out of it, what do these terms mean? Lecital comes from lecithin. There's that substance within the yolk that is called lecithin. So I'll write that here, L-E-C-I-T-H-I-N. So based on lecithin being in the yolk, lecital has to do with yolk, describes the yolk. So alecital, no yolk. So some cells actually do not have yolk, like extremely scanty yolk. Then microlecital. A cell is said to be microlecital when it has a small amount of yolk. Then, if the amount is beyond just being, extremely scanty, it is quite reasonable, but not so much, we could use the term mesolecital. And whether it is micro or mesolecital, the distribution of the yolk is another issue. So let me put that here now, distribution. So see this, there's quantity and that's distribution. So I'm trying to say now that based on the quantity of yolk, a cell may be, or an egg cell now may be described as being alecital, no yolk, microlecital, mesolecital, some quantity of yolk, macrolecital, plenty yolk. Now, if there is very little yolk, which is micro or mesolecital, how would the yolk be distributed within the egg? It will be something like this. So see this egg, see the yolk. In that case, we say that the yolk is evenly distributed. It is evenly spread throughout the egg. And that only happens if the yolk is not very much. So in that case, we say based on distribution, micro and mesolecital eggs are usually isolecital. So isolecital and microlecital, or isolecital and oligolecital, are not the same. They are not alternative terms. One of them describes the quantity of yolk, micro, oligo, whereas the other one describes the distribution of the yolk, isolecital. Iso, equal. So isolecital means that the yolk is spread all around. And I said that only happens if the yolk is not much. But if the yolk is much, which is macrolecital now, then the yolk will not be spread evenly all around. Instead, see the possibilities. You could have a case like this. This is the egg. What you have here is yolk. And then this is clear cytoplasm. A case like this is where you describe the cell as being telolecital. So this is a telolecital egg, telolecital. Then you can also have something like this. Everything here is yolk except that small disc on top, that small area on top. Everything else is yolk. This one is described as being discoidal. That's the distribution now. Then there's this other one, the last one here. In this case, this is yolk. That means there is a thin strip of cytoplasm around the yolk. So the yolk is concentrated where now? In the center. Because the yolk is in the center, central, you say that the egg is centrolecytal. So centrolecytal eggs, discoidal eggs, telolecytal. Telo, if you remember your cell division, telophase. Telophase is that stage where the sister chromatids would have arrived at the poles. Poles, that's the word, pole. So in this case as well, you have the yolk on one side, on a pole. So there's polarity in this kind of egg. We said it in the gametogenesis video that this area is called the animal pole. Why is it called the animal pole? There's no yolk there. And then where you have yolk, we said you call it the vegetal pole. So when there's distribution of yolk to one side of the egg, that's a polar egg. Now there's polarity. We say that type is polar lecytal. All right. Sorry, this is telolecytal. Okay, so this is discoidal, and that's what? Centrolecytal. And the terms are easy to remember. Central, center, discoid, of course, a small disc there called the blastodisc, and then you have telolecytal at a pole. All these three are under macrolecytal. So for macrolecytal, it may be telolecytal, it may be centrolecytal, it may be discoidal. So those are the different distributions that go with macrolecytal eggs, and this is the type of distribution that goes with micro or mesolecytal eggs. So looking at the eggs on the board now, macro, micro. What are the types of cleavage that can occur in these eggs? First thing I have to tell you now is, when you have a cell, an egg cell like this, the ovum, that is to undergo cleavage, it usually occurs by way of the formation of a streak that we call a cleavage furrow. So this streak that achieves the division is called a cleavage furrow. Now, the cleavage furrow is not known to cut through yolk. When you have a dense concentration of yolk, the cleavage furrow does not cut through it. So it means that cleavage furrow only cuts through clear cytoplasm or cytoplasm that has very little yolk. So in that case, if cleavage were to occur in this cell, how would it be? How do we expect it to be? Because there's a small amount of yolk everywhere, the cleavage that will occur here can be throughout the entire cell, can be on the entire length of the cell, and you have many cells like that. So this type of cleavage that occurs everywhere in the egg, because there's no dense yolk to stop it at any point, is the first one we would mention here, and it is holoblastic cleavage. So holo, from the word whole. So the whole cell divides. Everything about the egg divides. There's no yolk to stop the cleavage furrow. But in these other cases, you can't have the cleavage furrow going up and down. So see what will happen instead. In this telolecytal egg, for example, because there's dense yolk here, any cleavage that is going to take place will just take place at this point. This is where cleavage can take place. So there can be cleavage all through that. But for the yolk, no cleavage occurs there. What about this? Of course, all of this is yolk. So the cleavage can only take place at this small spot here. Then what about this one? You see yolk at the center. So any cleavage taking place can only take place on the outside here. These three conditions, where cleavage does not occur on the entire egg cell, but just a part of the egg because of too much yolk, is the second type of cleavage, and we call it meroblastic cleavage. So I'm saying now that there are two types of cleavage. The two types of cleavage are called holoblastic cleavage and meroblastic cleavage. Holo, complete. Mero, partial. Why is mero partial? Because there's plenty yolk and the cleavage furrow cannot cut through yolk. And why is holo complete? Because there's little or no yolk and the cleavage furrow is free to go around the entire egg cell. So, a summary of this: in terms of quantity of yolk, there are different types of eggs. Alecytal eggs, no yolk. Microlecytal, small yolk, very scanty. Mesolecytal, some yolk. Macrolecytal, plenty yolk. Isolecytal, of course, when there's little yolk, it will spread all around. Telolecytal, when there's plenty yolk at a pole. Centrolecytal, when there's plenty yolk concentrated at the center. Discoidal, when there's so much yolk that clear cytoplasm is just a small disc on top of the egg. And based on the yolk content, we said that cleavage that can occur in an egg cell can be of two types. If there's little yolk, little resistance to the cleavage furrow, then there will be cleavage in all directions. That's holoblastic cleavage. But in a case where you have yolk present, plenty yolk, then cleavage furrow has restrictions now. So in telolecytal eggs, there will be cleavage only at this point. In discoidal, just at the disc of clear cytoplasm there. And then in centrolecytal, the cleavage will be all around like this. Now, having mentioned that, let's go one step further in talking about the patterns of cleavage. When I say patterns of cleavage, we've seen the types of cleavage. There are only two types, and the two types of cleavage are holoblastic, complete, or meroblastic, partial, depending on your content. Now, for holoblastic cleavage, it has patterns. It may be bilateral, it may be spiral, it may have other forms. There are four forms actually. There's radial, yes, and then there's rotational. Beautiful. So these are the four patterns. These are not really types of cleavage. They are patterns of holoblastic cleavage, which means we may say for a particular organism that the cleavage that occurs in it is holoblastic. But it's not just sufficient to say it is holoblastic. You may want to say it is bilateral holoblastic, or spiral holoblastic, or radial holoblastic, or rotational holoblastic. So holoblastic has four patterns. Whereas for meroblastic, the patterns we have are bilateral, discoidal, and superficial. And interestingly, these three patterns occur in these three situations. Remember that in these three cases where you have plenty yolk, the cleavage must be meroblastic. We settled that part already. But which pattern of meroblastic? For telolecytal eggs, you expect a bilateral pattern. For discoidal eggs, you expect a discoidal pattern. Whereas for centrolecytal eggs, you expect a superficial pattern. So these are the three types, or the three patterns of meroblastic cleavage, and they apply to the three types of eggs that are macrolecytal. So you may want to take some time to go over this again before the break, so that when we return from the break, I'll comment briefly on these patterns, and then after commenting briefly on those patterns, I'll leave this number 2, which is what we've just done, which is cleavage, to tell you about blastulation and the formation of the blastula. Then I will also comment on gastrulation and tell you about organogenesis. So I want to ensure that there are just 2 videos to cover this aspect of our outline. So I'll see us after the break.